Hello, everyone. I'm Anne Rosser, and I'd like to welcome you to this meeting where we're going to hear about and discuss the emerging results of the Proof HD study. So before we launch into today's meeting, I just want to very briefly set it in context. So as part of a recent EHDN scientific strategy review, we decided that we should host a series of webinars to provide more opportunities for education and discussion, and that some of these should be devoted to discussing EHDN endorsed studies, of course, at an appropriate stage of their development. So this is the very first one of these meetings. So we invited Michael Hayden, CEO of Prilenia, to present the emerging Proof HD results, and he kindly accepted. I just want to say at this stage that many of the Prilenia employees are resident in Israel. So just to express that we're thinking of you at this time, and we really appreciate this webinar still going ahead. So after Michael's presentation, there'll be a Q&A session and then a panel discussion. And in the interest of transparency, it's important to say that Ralph Raumann, who's here to help to respond to questions and contribute to the discussion, is the European PI for Proof HD, and that I'm the European co-PI. So finally, I need to remind you that this meeting will be recorded. And at this stage, I'll hand over to Patrick White, a neurologist in Bonn and current co-chair of EHDN, who will host the meeting.
Yeah, thank you, Anne. Thank you for the introduction. Thank you, everybody, for joining us today online. I'll just briefly introduce you to the panel members and just let you know that the program is such that Professor Hayden will give a data presentation of what we're going to discuss today. Then we will have about 10-minute slot for Q&A that Anne Rosser and I will manage with questions that come in via the chat function. And then we will discuss the presentation and the results with the panel. I'm particularly thankful for Suzanne de Bott, from Leiden, who has stepped in at last minute for Sarah Tabrizi, who's unable to join today. And then we have Katrin Reetz, professor of neurology from the RWTH in Aachen, Hugh Rickards from University of Birmingham, and Bernhard Landwehrmeyer from the University of Ulm. So with that, Michael, I would hand over the screen to you for the presentation.
Well, thank you. Thank you to EHDN for this invite and giving us an opportunity to provide an update on Proof HD. This is, of course, still interim. Nothing's final. There are ongoing analyses occurring, but we welcome the opportunity to share the update as data is emerging. So thank you very much. I just want to start off by giving a thank you to all the participants in this trial, the whole HD community. It's a big study. The study participants, their families, the investigators, the site staff, and of course the steering committee, and the ongoing, which we won't talk about today, the ongoing high rate of participation in the open label study. Now, just to remind you, and not to go through too much detail, but there was extensive preclinical data that led us to believe that activation of this particular target was likely to have impact on numerous pathways, cellular biochemical pathways, that would have positive impact in HD. We also know that this is exquisitely mediated through sigma-1, because in all of these in vitro and in vivo studies that essentially demonstrated positive benefit in all these parameters and also are witnessed in the publications, if you delete sigma-1 or you develop an antagonist for sigma-1 in those experiments, the effects are completely nullified. So we know that sigma-1 represents a very important hub for these analyses. Now, this was a global 3, just to remind everybody, a global 3 phase 3 study. It had 499 patients, 250 per arm. It's a double blind. The primary endpoint was the mean change from baseline in TFC. The key secondary was CHDRS. And in the priority of secondary endpoints, we also had Q-motor, cognition, and a quality of life was an exploratory endpoint in this study. Now, already as part of the pre-specification for the study, we included the pre-specification of groups without antidopaminergics. And there was already data that alerted us to potential confounding effects of antidopaminergic meds. These were publications, some of them from different groups, from Roger Barker in Cambridge, from Joachim and others and published, where already it was shown that antidopaminergic treatment may be associated with some worsening of some symptoms, particularly impaired cognition. This was a subset of Enroll, and also this was also in registering cohort where there was significant, essentially progression associated with this. And this was also seen mostly in the neuroleptics, but also seen with chorea meds. And this was the reason that we included this as pre-specification of analysis with and without. Now, there were some limitations of these studies, and perhaps the biggest limitation is that these groups were not matched at baseline. So, for example, in the Tedroff study, the patients in the antidopaminergic group, and this was from registry, already had baseline decrease in TFC and some decrease in other parameters. They did rigorous analysis for covariates, but we were still concerned. And also, and therefore, we actually went recently to Jeff Long to ask him to see if it was possible to analyze the Enroll database to just get more information on progression of HD and the use of antidopaminergics. And I want to thank Jeff for his contribution, and Jeff will briefly present the results of this study that are ongoing.
Thank you, Michael. So we looked at different types of medication, antidopaminergics and antidepressants. This work was sponsored by Prilenia, and the goal here was to examine the association of the medication with HD progression in an early manifest population. So we queried the Enroll HD database using clinical and medication data. And in addition to looking at the antidopaminergics combined, we also looked separately at neuroleptics and VMAT2 inhibitors. The population is what traditionally has been called early manifest. So we have CAG equal to or greater than 36, age 25 or greater. We were looking at change over time. So part of the inclusion was to have people who had at least 2 visits in the database. These were all motor manifest as per the DCL. TMS was greater than or equal to 20, TFC greater than or equal to 7, and the independent scale was less than or equal to 90. So we focused on a binary coding of medication at each visit. So when we say people were exposed to medication, this means that they were on a medication for all of the visits throughout the duration of their time series. And when we say that they were off, then these are separate people who were off of medication for the entire time series. So there were 4 different exposure, non-exposure groups. As I mentioned, one was the antidopaminergics. That was the largest group because it was combined. It was a combination of 2 and 3 there, 1706 with a total sample size. Neuroleptics, VMAT2 inhibitors, antidepressants. And the time metric here is year from baseline up to 6 years. And the outcomes, we had the UHDRS variables of total motor score, symbol digit, Stroop word, total functional capacity, and the composite UHDRS, CUHDRS there. In addition, we looked at several motor scales. Some of these are from Karen Marder's work, Korea subscale, bradykinesia. We had both the scale and the individual item, dystonia, rigidity, ocular motor. Then we had gait and balance and hand movement. Next, please. So since this is an observational database, Enroll HD, there was imbalance at the first time point when people entered the study. So to attempt to adjust for this, we used something called propensity score matching at baseline, and we used the caliper here, which just meant that when we were matching, we were looking to find people that were a reasonably good match to individuals who were on medication. So after the matching, we used something called linear mixed modeling for group differences and rate of change. And again, we're looking at differences in rate of change over time. This was used after matching, and we used a particular type of model here that controlled for imbalance that persisted after matching. So the primary target of inference was group slope difference, and we did perform a sensitivity analysis, but I really don't have time to go into the details today. So here is an example of baseline imbalance before matching, which is on the left, and what occurred after we matched, which is on the right. On the horizontal axis, we have the standardized mean difference between the exposure and non-exposure groups. On the vertical axis, we have the variables. The very first one at the top there is distance, which is the distance between the propensity scores of unmatched and matched. And as you can see, under matching, of course, you have much more balance between the groups at study entry. Next, please. So here are the results. This is for ADMs, and this is for the UHDRS and variables. And I'm showing here the predicted curves with the data on the left, just so you know that there's data behind these prediction curves. And on the right, we have the same prediction curves, but without the data, so that you can glean a little better what the findings were. So all of the variables here had a significant difference in rate of change for the exposed and non-exposed, except for total motor score. And you can see on the right there, the red curve is for people who are on medication, and the blue is for people who are not on medication. So as you can see, there was a faster rate of progression for people that were on medication, though there was a very good balance at time zero, which was study entry. Next, please. So here are the motor subscales. Again, on the left, we have the curves with the data. On the right, we just have the curves. And in this case, there was a significant effect for chorea. But if you notice in the upper right there, those on medication had a slower rate of progression than those off of medication. And for bradykinesia, gait balance, and hand movement, it was the opposite. There were significant effects, but now we had those who were on medication having a faster progression than those off medication. Next, please. So we don't have time to show you all the results. I'm just going to summarize here. For the ADMs, for neuroleptics and VMAT2 inhibitors combined, we had worse rate of change for those on medication for the SDMT, et cetera, up there. Neuroleptics and VMAT2s showed essentially the same results, except that for VMAT2s, there was a larger difference than neuroleptics. And I'm showing 6 year results, and there were similar findings for 3-year results. For the motor subscales, there were a bit of difference here. For the VMAT2 inhibitors, there was significant worsening for rigidity, which was the only exposure that showed that. But there was no difference for chorea, or there was actually better progression for chorea under ADMs and neuroleptics, as you can see. Next, please. Then regarding antidepressants, there were no significant exposure differences for any of the variables. And you can see over there on the right for CUHDRS, those curves are basically one atop another. There are some separations you can see for Stroop word and total motor score, but this was not significant. Next, please. So what can we say? Well, ADMs are associated with accelerated HD progression. I can't say that this is causal. The sensitivity analysis, which I can't get into, was a bit ambiguous. And there were some unmeasured potential confounders here. We didn't take into account lifestyle like diet, exercise, substance use, or comorbidities, such as psychiatric disorders, diabetes, et cetera. And finally, of course, we didn't have a manipulation, so there was no control over the timing of the medication. People came into the study already on the medications, and medication must precede accelerated progression in order to establish causation, which we did not have here. Next. Oh, and then thanks to Michael Hayden, Paul Goldberg, Michal, EHDN, and especially to the participants and family members of Enroll HD who made all this work possible. Thank you.
Well, thank you. Thank you, Jeff. I think this helps us to think about our own data. Clearly, we've overcome the issue of matching, and still the same findings of increased progression associated with neuroleptics and VMAT inhibitors was found during Enroll in a much larger study. As you looked at our study, the study was well balanced. We actually baseline randomized for neuroleptics. We had career meds also not randomized at baseline, but as a sensitivity factor in the analysis pre-specified, and there was somewhat more in the predopidine group. There were a few people who were on more than one, and we don't have time to go through this today, but this becomes a discussion point. But as you can see, there were about 99 people on placebo who were on no ADMs, and 79 in predopidine. Much less power to see effect, but still some group of some standard to see the effect in this group. Safety data I won't go into, but just to say we have 1,500 patient years exposure, and remarkably, the safety profile is comparable to placebo. This is just just looking at the common related safety from placebo and predopidine, and essentially no difference between the two. And this also goes to treatment emergence adverse events. So the efficacy analysis, you will recall, we've presented some of this. Today, I'm going to focus primarily on the data excluding antidopaminergics and looking at some of the key endpoints here. And what you can see here, the primary endpoint, as we've shown before, and this is looking at the mean change from baseline in all patients in the study. And what you can see all the way through, there was no, this study did not meet its primary endpoint all the way out to week 78. When you start, and that was TFC. However, when you start looking at patients off antidopaminergics, you do see some, firstly, placebo goes down as expected, and you do see some stabilization, but this was not significant associated with this. Now, the secondary endpoint, cUHDRS, I won't go through what this measures, but of course, this is TFC, TMS, and cognition, and I'm going to present, just summarize some of the data. What you all know, in Huntington disease, all of these parameters show continued decline over time. This is just looking at cohort and track HD, where you see the decline in cUHDRS. And when we looked at our data, which what we saw is that not only was there no decline, but till about 1 year, there was improvement from baseline. So this actually showed improvement from baseline up to week 52, and then it stabilized with still differences, and above is improvement. On the right, we also want to show the mean difference between predopidine and placebo. And you'll see this in other slides. Going up means it favors predopidine. Going down means it favors placebo. And what you can see all the way along is that the cUHDRS of antidopaminergics favors predopidine over placebo. When you look at the actual extent of this in the classic and important Schobel paper, where the consensus was that slowing in decline about 20 to 30%, 0.2 to 0.3 is defined as clinically efficacious. And you can see here the numbers here are well above those for many parameters, suggesting this may be clinically meaningful. And if you look at the forest plots, just the big question with cUHDRS, and the regulators always want to know this, is this just dominated by one? And what you can see at every single time point, this is contributed, and going to the right over this line favors predopidine, the left favors placebo. For example, here, 26 weeks, there's the cUHDRS. And all of these, each of these 4 components contributes to the overall benefit at every single time point. So this is, of course, it's suggestive that these all contribute independently. It also, the regulators do like, and certainly you want to look at individual responses. What we were perhaps surprised was not that there was stability, but in some there was improvement in this. And so we wanted to also look at what the relative responder analysis was. And here you can see at every single time point, all the way to week 78, predopidine has improvement. The relative increase versus placebo is significant. The low at 38 weeks, the high at 104. But essentially, there's improvement from baseline in predopidine over placebo. Looking at some other functions, again, TFC, as we've spoken before, we don't have to go into it, but there's stabilization, but this was not significant. So there are trends to this. Of course, when you look at the mean difference to placebo, again, favors predopidine, but none of this reached significance. In Stroop, which was a key in cognition, again, what one saw is not that they were in placebo, you saw it go down as expected, and in predopidine, now you saw improvement from baseline, and that improvement continues all the way out to 78 weeks. In other words, it stays, never goes below baseline, all the way out to 78 weeks. So there's no progression, no worsening of SWR on treatment, and some improvement. Q-motor, of course, very important, and everybody knows that Ralph's involved, and it reduces inter-rater, reduces side effects, and this became a key secondary for us. And here you can see just some of the data. There's many measurements, but they're all mirrored by this. And what you see here, again, for placebo, as expected with Q-motor, this is looking at finger tapping ROI. You see improvement from baseline and actually stability all the way out to week 78. And here you can see with the standard error, you see the difference compared to placebo, all of this favoring predopidine. This is just another measure, just to show this wasn't, again, a single measure. It was seen in all measures. And associated with this, there's also, for example, here, change from baseline to week 78, looking at the correlation between Q-motor and cUHDRS. And again, these were highly significant. So the changes in correlation actually were correlated to the changes in cUHDRS all the way out to week 78. So this, of course, it's sensitive, Q-motor. It replicated findings we had in PRIDE, which I won't go into now. And of course, we know that Q-motor from TRACK-HD and others has been correlated with the TMS, TFC, and other measures. Quality of life, of course, so important. What was the impact of quality of life? And for this, we used the HD quality of life that recently was reported. It's had some validation by Eileen Ho and others, and it's directly relevant to the lived experience and consistent with patient-centered voice of patients as voiced by the FDA. And here what you see again is that totally independent measure, quality of life goes down over placebo. And here what we see is complete stabilization of quality of life, preserving the same quality of life in patients up to week 78. This was driven primarily by a few domains. These were significant. This was worries. The predopidine, this is improvement, so they did better. They were a little less worried. And also physical functional, it has all kinds of questions, also showed results that were above placebo all the way through to week 78. So, of course, for HD, We know that there's predictable decline in these factors. Improvements in these measures part of the natural history, not normally observed. And unexpectedly, these patients on pridopidine showed either improvements or stability on these endpoints of regression, function, cognition, and motor up to 78 weeks. Now, we wanted to go back. As everybody knows, there have been other studies that have looked at pridopidine. There have been 4 studies that have been looked at: PRIDE, MERMAID. This is actually HEART, PRIDE, MERMAID, and PROOF. That had adequate treatment, that were double blind, included the same target population, and had this particular dose. We, as a small, tiny team, this was done independently and externally. We just want to give you some of the results from this. These were the statistical models they used, which I won't go through now. But as we looked at this, these were the numbers. Now, in the whole study, in all the studies, these were the total numbers, but if you looked at patients off antidopaminergics, these were decent numbers now, significantly above what we saw in PROOF, and we wondered if there was more power to detect an effect. So for TFC, of course, in all patients in the study, what you see again, they deteriorate in the placebo in blue. Purple is pridopidine, no significant difference in all patients. And if you look at the mean difference, really very close to baseline, maybe favoring pridopidine in all patients. But when you go to patients off antidopaminergics, now you see, and actually this reaches a post-hoc significance, and certainly here it favors pridopidine all the way through. So we begin to understand that some of the issues with the prior studies is that they were never looked at off antidopaminergics, particularly of the studies 26 weeks and further. And what you can see here again in the pooled analysis, essentially these were, depending on which weeks, for example, let's just take week 26. Here you can see these are 3 pooled studies. All of them contribute to the data here in each of these particular studies. And this is significant all the way out to week 78. If you look at COHDRS, again, you see the same thing, that there is no data seen in, no improvement in patients on antidopaminergics, perhaps a little bit benefit here, but not really significant. And then when you look here, now you see off antidopaminergics, again, you see the improvement from baseline going up to week 52, and then it's stabilized. And if you look at the mean difference, pridopidine versus placebo, this is also showing benefit for placebo. And this is just looking at the pooled analysis, for example, for COHDRS at week 26, MERMAID and PROOF, essentially both showing the positive effect that weren't seen in patients on antidopaminergics. I'll just quickly show Q-motor. Again, what you see here, this is in all patients. You see some improvement, but essentially no real significance from placebo in all patients, perhaps at week 78. A mild improvement, but then when you go to patients off antidopaminergics, again, you see a much better differentiation with a difference relative to placebo being much greater. So I think just to summarize, so we can leave time, there was no benefit on the primary endpoint in all patients, but pre-specified analysis, which excluded patients on antidopaminergics, showed consistent improvement for different endpoints, and these captured independent aspects of the disease with a nominal significance at multiple time points. This includes COHDRS, Q-motor, Stroop, and quality of life. The benefit was sustained up to 78 weeks, and for example, and all the parameters showed improvement from baseline for at least 1 year, while placebo showed the predictable decline. And when we looked at Q-motor as an independent measure, this also showed quantitative improvement and correlated with COHDRS. And then the integrated efficacy essentially supported this of antidopaminergics. The benefit was driven by 4 clinical trials. And again, the good news is that pridopidine was safe. I just want to share, because we have other trial that's ongoing in patients off antidopaminergics, and this is pridopidine for ALS. and there's some late breaking news here. The Act for ALS, NIH funded, submitted by Healy, and just announced last Thursday, essentially the Healy Center and NIH, government of the US, is now supporting expanded access to pridopidine for ALS for patients not eligible for clinical trials. In other words, they're advanced. And this just came out October the 5th. And I'll just show you, I don't think we have time to go through all these results. In the whole population, again, there was no benefit because this included very advanced patients. When you looked at early patients and fast progressors, there were significant benefits. These were also pre-specified for ALS-FRS, respiratory, quality life, quantitative speech, and we now have encouraging results for survival, which I would show you. So here's, for example, in the fast progressors for pridopidine, what you see is significance. Here's pridopidine, slower progression compared to placebo starting at 8 weeks. And what you can also see here through oral analytics, there's an improvement in speech, by the way, in the whole analysis set. So this was significant speaking rate, articulation rate. Oral analytics has been accepted and got breakthrough designation from the FDA, and speaking rate and articulation rate, even much smaller numbers, showed post-hoc significance all the way through associated with this. And let me just show you survival, which is just very recent and relevant. So this was a double-blind study for 24 weeks. Then everybody went on to pridopidine. And these are Kaplan-Meier curves generated by Mass General and the Healy Center. And so this was the end of the double-blind study. One parameter is 50% survival. And what you can see here in the delayed start pridopidine group that was on placebo, they had a mean 50% survival of 300 days, whereas in the treated group on pridopidine all the time, it was increased now to an 85% survival. And then if you go on further, that there was a 10-month increase in survival in the ALS group, meaningful clinically delayed treatment effect in those who were on pridopidine all the time. So I want to just thank the people who conducted this study, HSG and TFS for excellent execution, EHDN for endorsing the study very early, and numerous partnerships with the various family-based organization, HDSA, Helpful HD, HDO, IHA, MENA HD, LRH, and of course, I'm deeply grateful to my colleagues at Prilenia. And just a moment on them, because, and particularly my colleagues in Israel, this has been a very heartbreaking and very difficult weekend. I posed to them that we could cancel this talk because they were still helping to get slides together. This was rejected by my colleagues in Israel. For many of them, their partners, their children, their relatives have been called up. They all have stories of people close to them who either have been killed, injured, or taken captive as hostages. And yet, despite that, they've continued to show amazing courage, amazing resilience, have been very inspiring, and their commitment to this community and the HDN ALS community has stayed steadfast, and they've remained very present all the way through from this weekend, from Saturday, all the way to today. So I'm deeply inspired by their resilience, and I want to thank them particularly. So thank you.
Thank you very much, Michael, for first, of course, for a very clear presentation of clearly complex trial results, but also for your final words, putting everything into perspective, and also special thanks to everybody here. And everybody is very much in our thoughts. I've been following this over the weekend, and I'm also very disturbed. There has been, but we'll in the spirit of what you just said, we'll continue with this discussion, and it's a lively discussion. So I've tried to follow it in the corner of my eye, so we'll only discuss it a very few minutes because I want to give 20 minutes to the panel discussion. So one question was if the people on antipsychotics were why were they? It's a question you've asked yourself, I'm sure too. Why were they included in the PRIDE study still, given all the previous results?
Well, in PRIDE, I mean, I would say the data was suggestive. When PRIDE occurred, that was 2013-14. None of these studies were published.
Mm-hmm.
So the Barker study came out in 2020. The Tedroff study came out in 2015. So these weren't published. We had no real indication, but we worried about it. And so we baseline randomized also for the antipsychotics or the neuroleptics in PRIDE, as we did in PROOF. So this is something I think we're learning as we go along. There's many questions that we haven't answered. We're not making any recommendations. We have numerous studies to go through. There's never been a prospective study of neuroleptics in HD. No double-blind prospective study ever, yet it's used off-label. We know that I developed and took the Austedo product to development. I led that. For Austedo, we never had to do anything except show that chorea was decreased at 12 weeks.
Okay.
We got approval for that. So there was no long-term study. All of this is emerging now, for which we are really trying to understand.
And what was the important, as a follow-up to this, was the analysis of the of excluding the patients on ADMs, was that then planned in 2013?
No, that was post hoc. We didn't know that. We did that. Also remember, in PRIDE, we had 33 or 34% of patients on neuroleptics. In PROOF, we had 60%. So the number of patients was less. And so in PRIDE, you still saw the effect on TFC because there was much less effect. But if you exclude ADMs for PRIDE, as you saw in some of these, rather fast, the improvement Q motor and, for example, TFC improved. You got better data, but it was a much smaller proportion.
Another direction of the discussion is teasing apart, of course, the dopaminergic or anti-dopaminergic effects and the S1R effects. One drug that came up was benzamides or sulpiride, which is a direct activator of the sigma-1 receptor. Did you have enough patients of that in your study to look at that separately?
No, very few patients on that. But again, I want to go back and look at that specifically, even a few patients. So for example, we are still going back to look, there were 14 patients that had had more than one antidopaminergic. In other words, either chorea meds or 2 neuroleptics. And we're still looking. It'll be anecdotal because there's not enough patients, 14 patients, but we are looking to see how they did. These are ongoing analyses that we're not quite ready to talk about yet.
And does the clear interaction with the dopaminergic with the anti-dopaminergic drugs suggest that or put into question that that the effect you're favoring the sigma-1 receptor? Could you comment on that?
Yes, and in fact, no, not at all. We know that sigma-1, and there's a lot of data from the past, that sigma-1 activation releases dopamine, for example, in the cortex, and also has impact on D1 and D2 receptors downstream. So if you either block dopamine secretion or decrease dopamine, that could have some mitigating effect. It also could block. And there's one other point to note. Pridopidine is a CYP2D6 inhibitor. It's excreted by the kidney, so you don't get high levels of pridopidine, but initially it's a CYP2D6 inhibitor. Exposure after 7 days, even in poor and metabolizers, is no different. But particularly the chorea meds, the VMAT inhibitors, are all metabolized by CYP2D6. And you would expect then you might get higher levels of these drugs. And certainly we know already from, if you just go to the label, the regulatory labels, both in Europe and the US for them, it talks about some of the dangers of higher levels. So we think we don't have absolute mechanisms. It's not fully understood, but we have a rationale. That doesn't explain how the data that Jeff Long saw or people have seen in the past on placebo, these patients are not on predopidine, you see worsening. And this still needs to be understood, the mechanisms associated with it. I don't think we know.
Okay, I have one. So the other questions go into something that we can use, stuff that we can use in the panel. But one question from the patient community is, do the data you presented here prompt you to plan another trial?
No, we're seeking advice. We believe that we have compelling data that's consistent, that's across endpoints and time points and measures independent aspects of HD. Okay. So we're going to seek advice, and we will take the advice we need and then make those decisions. We know that we are not aware of any drug that's ever shown results like this in any subset of patients. And so we're encouraged, but await further analysis of more data. It's still ongoing to understand much more.
Thank you very much. With that and with the time in mind, I will open the panel. I've introduced the panelists already, and I will maybe then ask the members for their just first impressions. Maybe Bernhard, I can start with you, Professor Landwehrmeyer from Ulm. Maybe your first impressions on this.
Sure, pleasure. And I think the data that Michael presented give us an impression of the complexity of the ongoing data analysis. I believe it's an interim report, and we need to await the final analysis. However, I think it's remarkable that there appears to be consistency in the findings concerning the phenotype in the absence of ADMs, and I think that's an interesting aspect to follow up on. I do have one question to Jeff Long's analysis, and maybe you, Jeff, could expand on this aspect. Did you, in your analysis, consider dose of the various compounds, or is that something that is still out to do?
Thanks for the question. Yes, that's still something out to do. We did consider that for this first run. We just coded things in a binary manner, but that it certainly is something to look at in the future. Thanks.
Yo, maybe Katrin Reetz now.
Yeah, thank you very much. Well, I also think that the data are very encouraging, and it might be a first step. I was really impressed, or so the first endpoint was not achieved, but I think we see a quite consistent improvement in different endpoints, and as it was also so pointed out in the presentation very nicely, these are also independent endpoints. So we have quality of life, we have cognition, and we also have the more objective Q motor. And I think this is a really good achievement, and hopefully a milestone for next medication and also clinical trials. I mean, we also know from other neurodegenerative diseases such as Alzheimer's, that, or also fetal ataxia, that also these first clinical trials that do not achieve the first the primary endpoint can be a door opener for further research and clinical trials. And this is really encouraging, and so we hope the best for further analysis and further research and trials.
Yep, thank you very much. Susan from Leiden.
Thank you very much.
The sound is not very clear.
Sure.
Now it's better, yeah.
Maybe thank you, Michael.
Can you go closer to the microphone, Susan? I think that would be better.
Did it work? Is this better?
Yeah, give us some more sound. So.
Thank you for the interesting results, especially also from the natural cohort from the enrolled HD. I think that's.
I'm sorry, Susan.
I'll fix it, and you go.
Yes.
Okay.
Maybe we move.
Rickard,
We move to
you can.
Hugh. Yeah.
Hugh.
Hi, just a couple of things. I guess first thing is, it's a message to get out to our community. If Jeff's results bear up, is we've got to think really carefully before we prescribe neuroleptics to our patients altogether. That's a potential revolutionary change and a message that we need to get out. The proof pride overall predominant findings are really interesting. I'm really pleased, Michael, that you've done that overall analysis. I know you had not much time to go through all the slides. Towards the end of the slides, when you're doing the sort of meta-analysis of all the trials, the Pride and the Mermaid, it seemed like you dropped out the Pride study from some of them, and I wasn't sure why that had happened. First of all, am I right about that? And if I'm right, what was going on there?
No, you're right in the sense that Pride did not measure SWR, Stroop.
It's only the Pride data was in all the other ones, but not in the Stroop.
Everything, but just because they didn't measure Stroop. We've also done, if you look at the appendix for the Scoble article, they show 3, and we've got this, I just didn't show it. We've done the CUHDRS similar to the way Scoble did this in the appendix, which also showed, just looking at TFC, SDMT, including SWR essentially, but looking just at the 3, TFC, TMS, and SDMT, and that shows almost as much sensitivity as the 4. And we see very similar results, but I didn't have time to go through this today, but I will talk a bit more about this at HSG. But yes, the only reason people were, as trial was excluded, if it didn't have a measure that went into that analysis.
So I've got kind of one more short question, if you like, which is, obviously these are all post hoc findings, this is a weakness, but they're quite consistent post hoc findings, which is a strength. How are regulators going to look upon this? If you go to regulators with this stuff, are they not going to come back to you and say, this is post hoc, you've got to go back and do it with a proper a priori trial?
Well, I have no idea what regulators are going to say, but just remember, 10% of all drugs have been approved that did not meet their primary endpoint. Okay. This is routine. This year, for example, it's 4 out of 40. There's an article in JAMA that goes through this. Not all these studies were post hoc. Some of them, so we can go through, but it's a mixture, and I have no idea what regulators will say. They look at risk benefit. Of course, the risk here with this drug in terms of safety is acceptable, but I have no idea what regulators will say, and we still decide. We're still analyzing data.
Okay, thank you.
Thank you. So Suzanne, how is your microphone doing?
Let's try. Can you hear me better now?
Excellent.
All right. Sorry for that. I was just commenting on Jeffrey Long's presentation, which is very illustrative for to see how the natural history is with and without antipsychotic drugs. So I was wondering whether in the proof data you also looked at the group which was on antidopaminergic drugs compared both placebo and on predopidine. So, and I'm asking this because I'm interested to know whether this is actually an interaction between the two, predopidine and antidopaminergic drugs, or is it another effect? So I'm interested in the other group. It's smaller, I think, but did you look at that?
we're in the process of generating all that data, but what we see, I'll just give you the highest, is that essentially in the presence of antidopaminergics, the effects of pridopidine are essentially lost or markedly diminished. So, and we're still looking at particular drugs and dosage. And we again don't have that data complete yet, but we'd like to know whether, for example, the labels of the drug, if you go to them, you will see that in the face of CYP2D6 inhibitors, the label from the FDA says you shouldn't use pridopidine, or you shouldn't use tetrabenazine above 50 milligrams. You shouldn't use Austedo above 36 milligrams. Gives very precise guidelines. And certainly this is a CYP2D6 inhibitor. What we don't know, and we'd like to know, is we look at our patients who were below those, did they have the same effect? We don't have that data yet, but this is very important data to come.
Yes, because I think we discussed before, Michael, about some of our patients who seems actually have like interactions. For example, when they went from the first study into the open label study, and it seemed that when they well, we don't know, we're blinded, but it seemed when they crossed over, especially in the beginning, they had more problems with akathisia and other, well, sudden worsening of HD symptoms, which might be an interaction between pridopidine but of course we are blinded, so we can just have a guess. So that would be interesting also to look at this group who made the switch.
Yeah, we will be looking at all of this, Suzanne, and appreciate that perspective. We want to learn that too. Yeah.
Yeah, great. Although it's very, of course, small numbers. That's always with post hoc.
Yeah.
Okay. So there have been a lot of activity now in the Q&A section. So I'll switch back to that if that's okay. There was one question on. Do you have an idea or feeling for long-term effects, given that some of the effects seem to be tapering off after time?
Well, we have data up to 78 weeks.
Mm-hmm.
And when you look at that, essentially, every single endpoint is improved over placebo for everything we've looked at, off antidopaminergics. So it's each time I'm talking about this. So we don't know. The study's only gone 78 weeks. We have open label that's just started. People have reached 26 weeks, and we will learn something from that. But again, it's not double blind. So we only have data that's double blind up to 78 weeks. There's data from open label from other studies, which did continue to show significant benefit of 5 years with total motor score and TFC, and we've published that. But those were tiny numbers, and I wouldn't say it gives us any further reassurance. So I would say right now we don't know the long-term effects, although at certainly 18 months, we're seeing a mean difference in every parameter improvement over placebo.
Okay, and the opposite question is also there. If this is particularly useful in early stage patients, do you have any thoughts on giving the drug to pre-symptomatic patients, given that there is a discussion about?
Yeah, of course, that's the golden question. These patients are usually not on antidopaminergics and not on career meds, and very early HD, potentially asymptomatic or patients that are pre-HD represents a crucial question, and we'd love to be able to answer that for a safe orally taken drug. Let me just make one other point. For us, universal access is very important. We are hoping that essentially the studies that are focused on knockdown will be successful, but even successful, the access to that technology will be limited. Hewish provided some data on that, and an oral drug that can be safe and tolerable and available globally is still, in general, something we should aspire to.
Thank you. So Bernhard has raised his hand. Yeah.
Yeah, and Michael, I was curious whether you could extend on one piece of data we saw by various measures a dip at 65 weeks. Do you have any idea what that may be, what the reason may be for this observation?
We don't. However, this was in the middle of the second phase of COVID. 25% of patients in this study had COVID. So it wasn't all that imbalanced between the affected, between people on active drug. So I would say the decrease at 65 weeks still remains unsolved for us, but we know it's due to COVID. The reassuring thing at 78, it starts coming back again. And even at 65 weeks, you're improved over placebo, but not significantly. There is a difference. So I don't think we have an answer for that.
Yeah, just a quick comment to that, actually, Bernhard. I mean, it is interesting, though. I guess that's the particular strength of quantitative measures, that it doesn't happen on the quantitative measures. Just to make a little comment on that here, because we do see that, obviously, as you know from many clinical trials, that clinical scales have quite a bit of variability. However, I think given the composite data set that we have looked at here, the joint studies and also the extension to more than 70 weeks, I think this is still an interesting lead. I mean, for me, I have to say, after, how many years now? I think it's been 15 years, Bernhard, that we were in Amsterdam to do the first investigator meeting for the MERM-HD study back then. I think, we've actually done our homework and examined this drug further, but all the clinical trials we've done, MERM-HD, HEART, PRIDE, all of them have shown some efficacy profile for this drug. You know what, in the end of the day, whatever the mechanism is, there is benefit to the patients, it seems, hidden somewhere. Definitely not worsening, which we learned sometimes in other trials. So I think it's an intriguing thing, considering the safety profile that we have here. Indeed, as was just discussed, potentially even to try this in prodromal premanifest where we don't have anything, and certainly want to have safety. So I think if looking at everything, all the data we have at hand right now, that the signals that we see here, even though they are post hoc, is it something we just don't want to pursue, even if this only works in part of the subjects? We see that in so many drugs. They don't necessarily need to be given to everyone with the disease in the end of the day. And you know what? I mean, I think we wanted to be very inclusive to begin with, and we all have discussed that. We didn't want to, to exclude a large subject cohort. Maybe it was a mistake scientifically.
Thank you, Leif.
That's where we are.
Thank you for this final perspective. We're already running out of time. I feel a bit sorry for that I didn't. So this was a very, very lively discussion. Thank you. Also, thank you to the panelists. And we've learned a lot. We're still, as Michael said, we're still learning from the predopitin trials. We're learning a lot about Huntington's disease. Hopefully this will translate into drugs. It will probably translate into how we manage the disease, especially with respect to the use of anti-dopaminergic drugs. And with that, Anne, I can give you the time is up. Maybe you can say some final words.
So just I agree with that, Patrick. Thank you very much. Thanks to Michael and Ralph and the Proof HD team. Thanks to all the panelists for giving up your time, and to Yuri and the EHDN team for supporting this meeting, and Patrick, to you for hosting it, and to everyone for joining. Apologies to anyone who didn't get their question answered. We will see if there's some mechanism for doing this at the end of the meeting. So,
and if you want to forward those questions, we could just try and communicate directly.
Fantastic.
Yeah, okay.
We'll do that. Thank you very much for that.
And Louise Beta is also saying, this is maybe important, that we didn't want to stigmatize anti-dopaminergic drugs. This is just a very good way, or a good incentive to think new about these drugs, but it's not a done deal, and people should not just stop taking their doctor prescribed drugs. Yeah, no, we continue to learn every year about this disease.
Yeah. So I think we've all learned a huge amount today. It's been a really worthwhile meeting. So thank you to everyone. And unfortunately, we'll have to finish there.